Dr. Terry, just, from a layman perspective, I'll be asking, sir, understand, I've heard the previous panel discussion, the whole cancer kind of, it's so humble experience. There's so much of unknown that is there. So just to want to ask you, CAR T is shown as, what should I say, it's been written as something like a miracle therapy. How do you characterize CAR T?
I would say that it is rare that a therapy comes along, really completely changes how you treat a certain type of cancer. And there's obviously other examples. I think the one example in ALL was the development, Gleevec, tyrosine kinase inhibitors for pH positive ALL and CML. That was completely changed the paradigm for how those patients were treated. I would say that CAR T cells, the experience to date has been similar. I think, as you heard today, the impact of CAR T cells is seen across all of the different treatment options for patients with ALL. There's now consideration around how CAR T cells fit with bone marrow transplant, how CAR T cells fit with standard therapy. So it really has been treatment that has really changed the way we think about treatment for ALL and for adult lymphoma. I would say the other thing about CAR T cells that is important to remember, it is, and is also really unusual, is that it is a completely different type of treatment than anything else that's used in oncology. Traditionally, when you think about treatment in cancer, it's radiation, it's surgery, it's chemotherapy. Now you have this, new treatment, CAR T cells, that is a completely new paradigm. So it really has, I think, changed the face of how we think about B-cell malignancies.
Sir, now there was a talk about dual CAR T. The whole science is advancing. What are the advancements that you are seeing? Because you closely interact with the whole biotech world. What exactly is happening in the world of CAR T?
I would say that CD19 as the first target for CAR T cells was very fortunate in some ways, because it turns out to be an ideal target for a CAR T cell. And again, we talked about it earlier in the panel, that the remission induction rate, the percentage of patients who get a CAR T cell who go into remission is 80-90%. That's unheard. So they're, extremely effective. The issue I think we're seeing is that it is going to be difficult in other types of cancer to achieve that same level of response. The one probably exception is multiple myeloma with BCMA CAR T cells. So what I'm seeing is that when you look across the biotech sector, et cetera, there's a lot of companies that are developing CARs targeting different antigens on different, different targets on the surface of the cancer cell for different types of cancer. So far, it looks like we're not going to see another CAR that's going to achieve a 90%, complete remission induction. So I think a lot of the advancements now in the field are more than just find another target on the tumor cell. There's other modifications to the T cell to make them more functional, for example, in a solid tumor, right? So I think you're going to see more and more of that.
So more than multiple target, it is like how to re-engineer the T cell is the most appealing research that is being done.
It's interesting. When we first started developing multi-antigen targeted CAR T cells, it made all the sense in the world, right? When you think about treatment of any cancer, one of the paradigms is that we use multiple agents. We typically don't treat cancer with a single chemotherapy, right? And part of that is because each of the different cytotoxic chemotherapy drugs targets a different part of the cancer cell, right? So the belief is that if you, not the belief, the data says that if you target the cancer cell in multiple directions, that it's harder for the cancer cell to become resistant to the chemotherapy. So it makes sense that you might be able to do the same thing with a CAR T cell by targeting multiple antigens, that it would be harder for the leukemic cell to become resistant. So it makes, although it's rational.
But what are the challenges in that?
The challenges are that we don't know the best way to actually go about doing that. You can target multiple antigens by putting two CARs in the same T cell cell. You can put single CARs in different populations of T cells. So you actually have one population that targets one antigen, another population of T cell targets another antigen. You can, instead of doing it at the same time, you can treat with one CAR, and then if the patient relapses, then you treat with the second CAR. So how to target multiple antigens most effectively is not yet known.
Sir, yesterday you also mentioned about Allogeneic CAR T. That is also been challenging. It can reduce the price of CAR T significantly, but it's still there are a lot of, in terms of remissions and all, there is challenges.
So obviously we talked earlier about the impact of CAR T cells in cancer treatment, and I made a comment that it is really a completely different type of treatment. One of the major differences obviously is that essentially each patient right now has their own drug made for them, right? So we know that each patient's products are different, etc. But with an autologous CAR T cell where you manufacture the CAR from the patient's own T cells, that is a completely different approach to drug manufacturing if you think about it. I mean it's never been done before like that. But obviously it creates challenges because it's expensive and it's complicated. So having an off-the-shelf, readily available CAR T cell product is somewhat of, I'll call it the holy grail. We all want that. We all know that that would be a major game changer. Unfortunately, the data so far with allogeneic CAR T cells suggests that the efficacy of allogeneic CAR T cells is not quite the same as autologous, largely seen not necessarily in the initial response to the CAR T cell, but in how long those responses last, persistence. That really seems to be the big issue, and probably involves some element of rejection of the CAR T cells that we're still trying to understand. Dr. Neelapu gave a great presentation yesterday.
Sir, one more, the ethical, it's not an ethical challenge, because the CAR T is so expensive, even in Indian context as well. Despite the price have been brought down. And you mentioned that 50% they show good remission and 50% relapse. And then they might need another immunotherapy, or if I'm not wrong, or BMT, bone marrow transplant.
Yes.
So how do you and you said that, you mentioned that we don't know which patients would relapse or which patient will show. For a treating doctor, what kind of challenge that you face?
I think it is an ethical challenge, right? So you just mentioned, the outcome. So 50% of patients will stay in remission, 50% of patients will relapse, right? I think most people believe that for an ALL patient that's refractory, whose cancer is refractory to other types of treatment, 50% patients staying in remission is pretty good. And I think most people believe that that's an acceptable outcome. Obviously, we want to be able to bring that number higher, but for right now, that's believed to be an acceptable outcome. I certainly think when you start looking at CAR T cells for other types of cancer, let's say that the durable remission rate is 10 or 20%. With that cost and complexity, there is some number where it becomes hard to justify the cost of the CAR T cell. So I do think that as we start looking at more complicated, more difficult to treat cancers with CAR T cells, etc., we're going to have to figure out ways, whether it's an off-the-shelf allogeneic CAR or some other, there are people who are thinking about generating CAR T cells in the patient without having to remove the T cells and manufacture. You can actually make the CAR T cells inside the patient. So there are a number of approaches that are being looked at to markedly reduce the cost from manufacturing autologous CAR T cell products. And I think for a lot of these other scenarios where you're looking at lower response rates, lower durable remission rates, the cost becomes not justifiable. And I do think that's why a lot of people in the pharmaceutical industry appropriately are looking at things like bispecific antibodies. So bispecific engagers essentially are a way of engaging the T cell to target the cancer without any manufacturing, because what it is it's the protein that bridges the T cell to the cancer cell inside the patient. You can make those a lot easier. The cost is lower. So rightfully, a lot of people are looking at those and saying, why would we continue to make CAR T cells when you have this other approach? So I think the field is going to have to struggle with that a little bit. I do think it's an ethical dilemma, and it is a cost prohibitive situation. I think, over the long term.
So you also mentioned that among the oncologists also, there are like two sets of people, one pro CAR T and one like little conservative on CAR T. I mean, like, how do you see that, sir? I mean, like...
Yeah, I think what you see in the adult oncology field, more so than pediatric oncology, because almost all children in the United States, I think the same is true in India, get treated at major medical centers. They're going to major medical centers that will probably have CAR T cells available. You know, the issue for lymphoma or for, you know, adult cancers where there's a much higher number of patients that are being treated at smaller hospitals, you know, centers out in the community that don't have CAR T cells, is that in the United States, often those patients will continue to get treated with, for example, the bispecific antibody for longer before they finally make their way to a major medical center to get to CAR T cells. And so I do think there's a lot of differences in practice depending on the center and their experience with CAR T cells. And I think, you know, we'll have to see how that looks over time.
Interesting, sir. And one more, like, you help ImmunoACT in developing the CAR T. And then how are you helping? Because the currently available therapies are so expensive. I mean, most part of the world, I mean, this therapy is not democratized enough. There are a lot of patients who are not able to get, I mean, at least the children, young people with ELL are like, show a very good remission and persistency. Personally, how are you helping, you know, this to reach to more patients?
You know, I guess I've been in the field developing gene and cell type therapies for pediatric cancer for about 30 years. And one of the things I always struggled with throughout my career was knowing that the complexity of these types of treatments were such that it was going to be very difficult if they worked. This was going back before the CAR T cell experience. I was developing cancer vaccines 30 years ago. I think I struggled a lot with trying to look to the future. If these therapies ultimately work, you know, how we were going to make them available, you know, to all of the children who would potentially benefit. And I honestly didn't see an easy path to that happening. So, you know, when I had an opportunity to visit, you know, India in 2015, when I was still working at the National Cancer Institute, you know, I didn't know, you know, what that visit was going to mean or what I would learn when I visited Tata for the first time and visited other medical centers in India. You know, where I sit right now, looking back 10 years ago, I guess was my first visit to India. I think I can safely say that one of the most satisfying things in my career as a pediatric oncologist has been being a very small part of the development of the CAR T cell program in India, because I do see it as a path towards making what is an extremely effective treatment for children with leukemia, making that treatment available potentially across the world, even out in lower middle income countries. And I'm aware of the fact that ImmunoACT is currently, you know, looking at collaborations in Mexico and other countries to be able to bring their CAR T cell to other countries. So again, I highly congratulate ImmunoACT on being able to do this. And for me, it's been one of the most satisfying things in my career. I can honestly say that.
So one more on the persistency of remission. How many years, sir, it's been like in your studies?
So the first patients were treated with CAR T cells probably around 2010, maybe even a little bit before that. The first case report of an adult lymphoma patient achieving remission was in 2010, first leukemia patients around the same time. So that, well, it didn't mean that that was the first report. So they were treated 2008, etc. Some of those patients now are out over 10 years, right? 10, 15 years. And some of those patients are still in remission. So we know that some of the remissions can be very durable, probably a cure. I mean, we always are careful saying that in cancer, but probably those patients are cured. We also know from experience with some earlier CAR T cells targeting HIV that some of those CARs persisted for 10 plus years. So we know the persistence can be not just measured in months, but can be measured in years. So I do think that once we kind of learn how to apply it effectively, how to select the right patients, maybe manipulating the T cells so that they persist better, etc. I think what you end up with, at least with the autologous CAR T cells, is a scenario where those patients have some degree of surveillance for their cancer for years, potentially. And, you know, that's pretty amazing with a single treat. So,
sir, I mean, during COVID, the whole immune immunity as part of science have received a lot of interest, and a lot of scientists who are like working on the immune system and its various ways, T cell have gained a lot of attention globally. Now where is the research heading, sir? I mean, now that COVID has died down, I mean it's not died down, but as little, where is the whole research going on now? Because it is tied up with both oncology as well as rheumatology.
Yeah, I mean, I think it is interesting. I mean, my favorite, system, the immune system that I've been studying for 30 years, it is true that with COVID, there's been a much broader awareness of the importance of the immune system. And remember that the impact of the immune system on cancer is not actually, the biggest impact hasn't really been CAR T cell. The biggest impact has really been with immune checkpoint inhibitors, right? Okay. So immune checkpoint inhibitors, Nobel Prize awarded to scientist Jim Allison, Kusu Hanjo in Japan, got the Nobel Prize for the identification of immune checkpoint inhibitors. So there's really two areas where the immunotherapy has had a huge impact, and CAR T cell right now has actually been smaller. The reason I think why CAR T cell has gotten a lot of press is because it's not just an immunotherapy, but it's actually a gene therapy. So it was the first approved gene therapy in the United States, technically. So it's an immunotherapy and a gene therapy. So I think it set the foundation for a lot of other treatments, even outside of immunotherapy. So we now know that gene therapy can be very effective for sickle cell anemia, thalassemia, rare pediatric genetic disorders. But, coming back to where's the research going? I think the foundation that was built with CAR T cells and with immune checkpoint has really created a completely new field. I think what you're seeing, for example, with the application of CAR T cells to lupus for rheumatologic diseases like lupus and rheumatoid arthritis, is you're seeing the next chapter really in this whole field evolve. And then in parallel, as I said, you're seeing the application of gene therapies more broadly across a whole spectrum of diseases. So, we had a discussion yesterday at the meeting. There was a presentation, a very interesting presentation from the head of the regulatory agency in India. And I think we saw the same thing in the United States that we heard yesterday, was that with the first CAR T cells, the FDA didn't really yet know what to do with gene therapy. And over the course of the last 15 years, the FDA has developed, guidance documents and very specific approaches to evaluating novel gene therapies. But it is so different from traditional drug development that it's a completely new paradigm even for the regulators. And so I think the research that's happening now is really built on the foundation that was created by the early experience with CAR T cells and immune checkpoint inhibitors in many ways.
So, sir, thank you so much for taking your time out and speaking to me, sir. Thanks so much.
My pleasure.
Sure.
Thank you.